issues, that was the live have been disconnected, so I am just revising the last topic and then continuing. Chargaff first parity rule means what I have told you about the Chargaff rule, that is the percentage of adenine will be approximately equal to thymine and the percentage of cytosine will be approximately equal to guanine. So I'm continuing from that. Chargaff first parity rule is property of double stranded DNA which states that the number of adenine and thymine nucleotide the number of adenine and thymine nucleotide and the number of cytosine and guanine nucleotide are the same within the duplex. It arises as a result of chemistry of double stranded DNA, which only permits adenine to pair with thymine and cytosine to pair with guanine. In contrast, Chargaff's second parity rule asserts that the same is also true within the single stranded. Means for the double stranded and single stranded, both the rules are same. However, unlike the first parity rule, the second is not exact. But this rule has been puzzled in a molecular biology as exact reasons are still not clear. In a double stranded DNA, the molar ratio of adenine to thymine equal to 1 and similarly the molar ratio of guanine to cytosine equal to 1. The sum of purine equal to the pyrimidine and the percentage of cytosine plus guanine does not necessarily equal to the percentage of adenine plus thymine. So now we will study the structure of double stranded DNA. James Watson and Francis Crick, working in a Cavendish laboratory at Cambridge University in 1953, took advantages of Chargaff finding and the X-ray diffraction data of DNA fibers obtained by Rosalind Franklin and Morris Wilkins, described the structure of the DNA double helix and X-ray diffraction data of a DNA fiber provided evidence about the helical nature of a DNA molecule. Watson, Crick, and Wilkins were awarded in 1962 by Nobel Prize for Medicine for discovering the molecular structure of DNA. Rosalind Franklin died of cancer before the Nobel Prize were awarded. Nobel Prize are not awarded posthumously. The Watson-Crick double helix structure describes the features of B form of DNA. However, there are many other forms of conformations of DNA, such as A, C, Z form, which are very distinct from the B form. The form that DNA would adopt depends on several factors, like the hydration level, means the how much water content is present, DNA sequences, what is the sequence of DNA, chemical modification of the bases, means which base is present, whether it is adenine, guanine, thymosine, the type and the concentration of metal ion in solution. So we will now going to study bDNA. The Watson-Crick double helical structure is referred to as bDNA, means the DNA discovered by Watson and Crick was bDNA. It is the most stable and predominant form under physiological condition. In this form, two long polynucleotide strands coil around a central axis to form a right-handed helix. Strands are antiparallel, that is, one strand is oriented in the 5' to 3' direction and the is uh, second is obviously the 3' to 5' direction. The strand interacted by hydrogen bond between the complementary base pair. The base G form three hydrogen bond with the C, and similarly the adenine form hydrogen bond, that is will be double bond with thymine. You can comment in the chat box whether are you understanding this or not. So the bases occupy the helix core and the sugar phosphate chain run along the periphery, minimizing the repulsion and between the charges of the phosphate. Since we know that a nucleotide consists of nitrogen base, sugar along with the phosphate, and by binding them we can keep the opposite direction, and so that the repulsion will be minimum. Now the sugar phosphate backbone of the helix are not equally spaced along the helix axis, and the grooves between them are not the same size. Instead, the intertwined chain create a major groove and a minor groove. Major groove is wide and deep, whereas the minor groove is narrow and deep. The site containing nitrogen 7 of purine and represent the major groove, while the other site containing nitrogen 3 of purine is the minor groove. Now the bDNA is characterized by 10.4 base pair per helical turn, and the axial height of one helical turn is 33.2 angstrom, or whether we can say 3.32 nanometer. The rise per base pair along the helix is 3.32 angstrom. The base pair are arranged nearly perpendicular to the helical axis with anti-conformation for all glycosidic bond and the C2 endo sugar pucker of all nucleotide. The resulting helix has a diameter of approximately 20 angstrom. And in alternative form of the right-handed double helix is aDNA. Means the alternative form of bDNA is aDNA. aDNA molecule differ from bDNA molecule in a number of ways. That is, the aDNA is characterized by 11 base pair, and have you remember what is the number of base pair in bDNA? If you remember, please comment on the chat box. The aDNA is characterized by 11 pair per turn, and the helix pitch is 24.6 angstrom, or we can say 2.46 nanometer. There is a rise per pair along the helix axis of 2.3 angstrom. Like bDNA, aDNA also has anti-conformation for all glycosidic bond, but C3 endo sugar pucker. Do you remember in bDNA what is the sugar pucker? That was C2. And by continuing the same line, the bDNA is thus longer, okay, and thinner than aDNA. Relatively dehydrated DNA fiber can adopt a conformation. Features of major group and minor group also differ. The right-handed helical DNA, RNA hybrid, and double-stranded RNA also adopt A-like conformation. The 2-OH in RNA sterically prevent double helical regions of RNA chain from adopting the B conformation. The helical parameter can be categorized into two general classes to describe the absolute and relative conformation in DNA. Base pair parameter, that will be means under base pair parameter there was translation and rotational relationship of bases in a single base pair. Means in base pair parameter we study the translational and rotational relationship of bases in a single base pair, and in a base step parameter translational and rotational relationship between two stacked base pair. Parameters such as propeller, twist, shear, stagger, and stretch are base pair parameter, whereas shift, slide, rise, tilt, roll, and twist are the base step parameter. Means parameters such as propeller, twist, shear, stagger, and stretch are the base pair parameter, whereas shift, slide, rise, tilt, and roll, twist are the base step parameter. We can understand these parameter by considering x, y, and z axis. Means we consider the three axes that was x, y, and z, and the x and y axis lie in the plane of the base pair, with the y axis lying along the long axis of a base pair. The z axis is perpendicular to the base of the base pair and parallel to the helix axis. Displacement of one base pair with respect to other along the x-axis, y-axis, and z-axis are called shift, slide, and rise respectively. Means the displacement of a base pair with respect to the x-axis. Under x-axis, the displacement will call shift, y-axis slide, and z-axis rise. Three angular parameters such as tilt, roll, and twist define the position and direction of one base pair relative to the next. Tilt is the rotation of base pair along x-axis. Roll is the rotation of the base pair around the y-axis, and twist is the rotation of the base pair along z-axis. We can remember it by Tx, Ry, and Tz. Now we will study the Z-DNA. Z-DNA is a left-handed double helical structure with two anti-parallel strands that are held together by Watson-Crick base pairing. The transition from B to Z DNA conformation occur mostly readily in DNA segment containing alternatively purine and pyrimine, especially alternate C and Z on one strand, and also the DNA segment containing alternate T and G on one strand. The existence of zDNA was first suggested by optical studies demonstrating that the polymer of alternating C and Z in one strand in a 4 molarity NaCl solution. The physical reason for this finding remained a mystery until X-rays crystallographic study of a self-complementary DNA hexamer that will be abbreviated as DCZ to power 3 revealed a left-handed double helix. Means aDNA and bDNA was right-handed helix, but now here zDNA is a left-handed double helix with two anti-parallel chains that were held together by Watson-Crick base pairing. zDNA is thinner 18 angstrom and bDNA 20 angstrom, and there's only one deep narrow groove equivalent to the minor groove in bDNA. No major groove exists. In contrast to bDNA, where a repeating unit is one base pair, in zDNA the repeating unit is a two base pair. This nucleotide repeats causes the backbone to follow a zigzag path. A zigzag path rise to the name zDNA. The glycosidic bond conformation alternate between anti and syn for anti-pyrimidine, rise syn for purine. Similarly, the sugar pucker alternate between C3 endo and C2 endo. C2 endo for pyrimidine, and C3 endo for purine. Means in zDNA, the sugar pucker will be C2 endo for pyrimidine containing nucleotide based DNA and C3 endo for purine containing nucleotide nitrogen base. zDNA can form in regions of alternating purine pyrimidine sequences like GC sequences form zDNA most easily. TG sequences also form zDNA, but they require a greater stabilizing energy. Formation of zDNA conformation is generally unfavorable. However, certain condition promotes zDNA conformation from bDNA conformation, such as negative DNA supercoiling, high salt concentration, and 5-methylated cytosine. Now I am just revising line by line, and you can make a short note on the difference between A DNA, B DNA, and Z DNA. So I am just slowly reading, and you can make a short note. So helix sense. So first of all, you write the 4 things on the upper line. That is, in first box, the title will be geometry attribute. In second line, A form. In third, B form. In fourth, Z form. Again I am repeating geometry attribute. Now write A form, then B form, and then C form. Sorry, Z form. So first heading is helix sense. So helix sense, please comment on the chat box whether you have written or not. So helix sense, in A form right-handed, in B form right-handed, and in Z form left-handed. Second diameter, in A form 23 angstrom, in B form 20 angstrom, and in C form, means the Z form, 18 angstrom. Now third one is pitch, that is axial health of one helical turn. In A form 24.6 angstrom, in B form 33.2 angstrom, and in Z form 45.6 angstrom. Now fourth one is mean base pair or helical turn. In A form 10.7, in B form 10.4, and in Z form 12. Axial rise of one base pair. In A form 2.3 angstrom, in B form 3.32 angstrom, and in Z form 3.8 angstrom. Now twist angle, that is rotation per base pair. In A form 33.6, in B form 34.3, and in Z form 60 degree by 2. Now base per tilt, that is 20 degree in A form, minus 6 degree in B form, and 7 degree in Z form. Mean propeller twist, plus 18 degree in A form, plus 16 degree in B form, and in Z form 0 degree. Glycosidic bond conformation, that is in A form NT, in B form NT, and in Z form NT for pyrimidine and syn for purine. Now sugar pucker, in A form C3 endo, in B form C2 endo, and in Z form C2 endo and C3 endo. Now repeating unit, one base pair in A form, one base pair in B form, and two base pair in Z form. Now major groove, narrow and deep. Narrow and deep for A form, wide and deep for B form, and flat for Z form. And in minor groove, wide and shallow for A form, narrow and deep for B form, and narrow and deep for Z form. That's all for the table. And now we are again going to learn a new topic, that is triplex DNA. So you have already heard about duplex. Now we will study the triplex, and there are quadriplex also. That will be the next topic. So, triple helical nucleic acid was first discovered by Felsenfeld and Rich in 1957. In certain circumstances, that is low pH, a double-stranded DNA consisting of homopurine sequence in one strand and a homopyrimidine sequence in complementary strand. Sorry. So, in some circumstances, that is in some low pH, a double-stranded DNA consisting of a homopurine sequence in one strand and the homopyrimidine sequence in complementary strand form a triplex DNA with either a pyrimidine rich or a purine rich third strand. The third strand DNA in a triplex structure follows a path through a major groove of a duplex DNA. The triple helix will be written as Pu star Pu dot Py. That is, 2 purine is going to form the third hydrogen bond with the pyrimidine, and the central strand of the triplets must be homopurine. The third strand bases make a Hookesten and reverse Hookesten hydrogen bond to a homopurine strand of a duplex. Typically, pyrimidine-rich third strand bind to the homopurine strand of the duplex DNA via Hookesten hydrogen bonding in a parallel orientation. That is, in the same 5' to 3' orientation as homopurine strand of a duplex, whereas the purine-rich third strand bind in an anti-parallel orientation to the homopurine strand of the duplex via reverse Hookesten bond. Triple helix can be intermolecular or intramolecular. In intermolecular triplex HDNA, the third strand is provided by one of the strands of the same duplex DNA molecule. Formation of both intramolecular and intermolecular triplexes depends on the several factors like oligonucleotide length, base composition, pH, divalent cations, and temperature. Triplexes can be built from RNA and DNA chain or their combination. Now we are going to study about Hookesten base pairing. So I'll make you understand what is the meaning of Hookesten. Hookesten base pairing is quite different from the standard Watson-Crick base pair. In Hookesten base pairing, the glycosidic bond linked to a purine a syn rather than an empty conformation. In AT Hookesten base pairing, there are two hydrogen bonds as shown in the below. And the N7O6 and N6 of purine, the atom that participate in the hydrogen bonding with the third DNA strand are often referred to as Hookesten position. The, the Hookesten base pairing between guanine and cytosine can be formed only if cytosine is protonated in the N3 position. The GC plus Hookesten base pairing, two hydrogen bond occurs as compared to GC Watson-Crick base pairing. Understand? Due to the plus charge in case of Hookesten bonding, in between the G and C there was 3 hydrogen bond in Watson and Crick model, but here in Hookesten hydrogen bond there will be 2 bond That will be Hooke's hydrogen bond between G and C. Now we are going to study the G quadruplex. The G quadruplex or tetraplexes or G4 structure are four-stranded secondary structure forming by one or two or four separate strands of a DNA and RNA molecule. Nucleic acid, both DNA and RNA containing guanine-rich sequences spontaneously fold into G quadruplex structure. The building blocks of G quadruplex or G quadrates, also termed as G tetrad, that are formed through a cyclic Hooke's hydrogen bonding arrangement of four guanine with each other. The planar G quadrats stack on top of one another, forming four-stranded helical structure. The formation and stabilization of G quadruplexes are dependent on monovalent cations, especially K plus and Na plus. The stabilization is due to the coordination of the positively charged cations with the electronegative O6 atom in the center channel of the adjacent stack G quadrates. The G quadruplex structure are topologically very polymorphic and can arise from the intra or intermolecular folding of G-rich strand. Intramolecular folding requires presence of four or more G-rich sequences in one strand, whereas the intermolecular folding can arise from two or four strands, giving rise to a parallel or anti-parallel structure. When at least one of the four strands is anti-parallel to the other, all parallel quadruplexes have all the guanine glycosidic bonds in an anti-conformation. Anti-parallel quadruplexes have both syn and anti-conformations. In vitro thermal stability of G quadruplexes depend on the features such as the number of G quadrats present in the structure and the length and composition of the loop formed by non-guanine bases. Now we are going to study the stability of double-stranded DNA helix. Double tap on the screen to like this live. So the helical structure of double-stranded DNA is stabilized by non-covalent interactions. These interactions include stacking interactions between adjacent base pair and hydrogen bonding minor. Means if we talk about double stranded DNA helix, stacking interaction will be the major interaction as compared to the hydrogen bonding. Hydrogen bonding is a minor interaction between complementary strands. The core of the helix consists of a base pair with stacked together through stacking interaction. These interactions include hydrophobic interaction and van der Waals interaction between bases that contribute significantly to the overall stability. The base stacking minimizes the contact of water with the non-polar bases. Since the bases are planar, the base pair are stacked nearly on top of one another. The typical separation between the planes of adjacent bases correspond to the van der Waals radius. So the typical separation between the planes of adjacent base pairs correspond to the van der Waals radius. The maximizes This thing maximizes the van der Waals interaction between the base pair. Internal and external hydrogen bonds also stabilize the double helix. The two strands of a DNA are held together by hydrogen bonds that forms between the complementary purine and pyrimidines. Two hydrogen bond in an AT pair and three hydrogen bond in GC. while the polar atom in the sugar phosphate backbone form external hydrogen bond with the surrounding water molecules. So I would like to repeat this. While the polar atom, that is the polar atom present in this nucleotide in the sugar phosphate backbone. So in sugar phosphate backbone, the polar atom is obviously the oxygen of phosphorus, and this will form external hydrogen bond with the surrounding water molecule. The overall energy of the hydrogen bonding depends predominantly on base composition, that is, all AT and GC bond combination of one base pair. On the other hand, the base stacking interactions depends on both base composition and base sequence. Some combination of base pairs forms more stable interaction than other. For example, a GC-GC new dinucleotide stack has a stacking energy of minus 14.59 kilocalorie per mole per stack pair, whereas the TA means thymine adenine stack has an energy of minus 3.82 kilocalorie per mole per stack pair. Once the DNA double helix is formed, it is remarkably stable. The individual interaction stabilizing the helix are weak, but sum of all interactions makes a very stable helix. So now we are going to study DNA naturation. So there are terms naturation, denaturation and renaturation. Naturation means the DNA is normal and a double standard. When we heat something like DNA to a particular temperature, it will the both the strand get separated and it became denatured. Now again, when both the strand form bond hydrogen bond between them, that will be renaturated. So let's start this topic of DNA denaturation. DNA denaturation is a process in which the double stranded DNA separates into two single stranded due to disruption of hydrogen bond and stacking interaction. That is, it is a process of separation of double stranded DNA strand, and the agent that causes denaturation is called denaturing agent. These agents can be physical like temperature, chemical like urea. Urea causes denaturation by acting as a chaotropic agent. Chaotropic agent, a new term is found. Chaotropic agent basically a molecule in water solution that can disrupt the hydrogen bonding network between water molecules. That will be called chaotropic agent. Again, I'm repeating, chaotropic agent are the molecules in water solution that can disrupt the hydrogen bonding network between the water molecules. If the temperature is denaturing agent, the double helix is said to be melted. Now the temperature at which the half of the double stranded DNA is denatured is called melting temperature, and that is denoted by Tm. Tm is basically called a melting temperature, and it is a temperature at which the half of the double stranded get denatured. So the high value of Tm is related to the high thermodynamic stability of DNA double helix. The DNA denaturation is a cooperative process. The denature basically it is a cooperative process in which one and more agents like temperature, chaotropic agent, these are dependent on each other sometimes. The denaturation process is accompanied by a change in the DNA physical properties such as optical density, viscosity, and band density. So, density, maybe band density, basically it is a property of something that will get float on the water molecule. And now the aromatic nitrogenous base present in a DNA and RNA absorb maximum of 260 nanometer, means the aromatic nitrogen bases absorb maximum wavelength of 260 nanometer. The absorbance at 260 nanometer is a consequence of a transition delocalized pi electron. This absorbance at 260 nanometer increases with the increased transition. When the bases remain stacked together in double stranded DNA, they interact via their pi electron cloud. These interaction decreases the pi electrons transition and thus absorbance. Unstacking of bases increases pi electron transition. Due to the diminished interaction. As a result, the absorbance at 260 nanometer increases. Denaturation of the double-stranded DNA, also double-stranded RNA, increases the absorbance or optical density at 260 nanometer. This increases in the absorbance at 260 nanometer is known as hyperchromic shift. So a term also came, that is hyperchromic shift. So what happened when the DNA absorb maximum light of 260 nanometer wavelength? This absorbance at 260 nanometer increases with the increased transition. When the bases remain stacked together, they compact together, they interact with their pi electron cloud, and this interaction decreases the pi electron transition and thus absorbance. Unstacking of bases increases pi electron transition due to diminished interaction. As a result, the absorbance at 260 nanometer increases and denaturation of the double stranded due to diminished interaction occurs, and this increasement of the absorbance level at 260 nanometer is called as hyperchromic shift. Now, this occurs because in double-stranded helix, bases remain stacked, whereas denaturation causes strand separation and unstacking. Because of denaturation, the bases become mostly unstacked and thus absorbance increases to 60 nanometer. Denaturation is also accompanied by decrease in viscosity and band density. Denaturation also accompanied means it also occurs in decrease when the viscosity and band density decreases. It also causes denaturation. Now we will study the factors at affecting Tm. The melting temperature is affected by the following factor. The Tm value or the thermal stability of double stranded depend on several intrinsic and extrinsic factors. These factors are Gc content. Remember, melting temperature get affected by Gc concentration, means GC content in DNA, ionic strength, and obviously the pH. So, factors affecting Tm. Melting temperature is a temperature at which half of the double stranded DNA gets separated, and the factors affecting this melting temperature is GC content, ionic strength, and obviously the pH. So, the double stranded DNA with a high GC content has higher Tm, and DNA with lower GC content means it is directly proportional to the GC content. As the number of GC and as the GC content increases, TM also increases. As the GC decreases, it also decreases. Now coming to the ionic strength. Tm is also dependent on the ionic strength of the solution. The lower the ionic strength, the lower the melting temperature. It means it is also directly proportional to the ionic strength. As the ionic strength decreases, steam also decreases. As the ionic strength increases, steam also increases. DNA is a polyanionic molecule. Each phosphate group in a DNA strand carries a negative charge, and this negative charge on both the strands repel each other. The magnitude of the repulsion is diminished by a presence of cation such as sodium plus and magnesium 2 plus. These cations interact with phosphate group and neutralizes their negative charges, and when the charges are not neutralized, the electrostatic repulsion decreases the Tm of the double stranded DNA. Now the third topic, and this will be going to the last topic of today's live class, and that is pH. Change in the pH also affect the melting temperature. The reason behind that is the protonation deprotonation of the bases due to change in pH. At high pH greater than 10, extensive deprotonation of the base occur, destroying the hydrogen bond potential and the denaturation of the duplex. For example, the pK of proton on N1 in guanine is typically 9.7. When the pH approaches to 10, N1 guanine is deprotonated because this proton participate in hydrogen bonding. Its loss destabilizes the DNA. Similarly, the extensive protonation of the bases below the pH 3 also disrupts the pairing. Below pH 3, some of the hydrogen bond except that participate in base pairing become deprotonated, and in their protonated form these pair can no longer form a hydrogen bond. So that's all for today's class. I'll see you tomorrow in the next live class where we will study the quantification of the nucleic acid. So thank you for joining my live session. Take care. Bye bye.